Hey there everyone! So in this video we're going to go ahead and take a look at the animals that are known as sponges. Alright, sponges belong to the phylum Porifera, which literally translates means pore bearer, and that's definitely one of the key characteristics that we use to identify these animals, is that their body contains numerous small openings that we call pores. Alright. Now we've actually mentioned sponges previously in lab when we were talking about the different grades of animal construction, and these were really the only animals that represent what we call the parazoan grade of construction, meaning that they're really just at a cellular level of construction. They have a lot of specialized cells, but they're not really organized into true and permanent tissues. Alright, so you can see in the screen right here we've got some wonderful pictures of some marine sponges. They are certainly most diverse and most abundant in a wide variety of marine habitats, especially in coral reef ecosystems. So you can see that a lot of them have these kind of slender, tubular shaped bodies, but as we're going to get into, they actually come in a wide variety of body forms, and we'll learn a lot more about that as we go through the presentation. Alright, so let's go back and let's talk about just sort of the basic construction of a sponge, and we're going to start with the different cell types that are responsible for making these bodies. Alright, so we can see here that there are kind of four key types of cells that are going to make up the body of a sponge, and you guys will have a chance to look at some of these and identify these cell types in some slides as you go through the lab activity. Alright, so first on our list here we have what are called porocytes. Alright, so this is really where the phylum gets its name from. Porocytes in simple sponges are these actual tube-shaped cells that are hollow. So there's actually a channel that passes kind of through the body, and the rest of the cell body is kind of made in a big circle around it. If you look at it in cross-section, as you can see over here, you can see that there's this little hole that goes from what would be the outside of the sponge body into the inside. Alright, so that's an interesting and very unique characteristic of this particular animal phylum, are these cells. Now, why would they need pores or holes in them? Well, that comes to the major role that this next group of cells have. Alright, so this funny looking name here is actually pronounced choanocytes. Alright, choanocytes, so a hard K, not a CH sound. Alright, so choanocytes, what are those? Well, those are way over here on the right side, lower right side of the picture right here. And these are really unusual and interesting cells. So they kind of have your regular cell body, nucleus, mitochondria, all the organelles. But an interesting feature of choanocytes is that there are these thin little blade-like extensions of the plasma membrane that form this ring-like structure referred to as a collar. And that's one of the nicknames for these cells. They're called collar cells. And here you can see that emerging from out of the center of that collar is a long flagellum. Alright, so what is this all about? Well, this is really all about how these animals are able to get the resources that they need from their environment. All sponges are aquatic organisms, and they rely on water. So what they rely on water for is, of course, primarily oxygen and access to food and nutrients. Alright, so the way that oxygen flows through a sponge body, and water flows through the sponge body, is that these choanocytes are all lined on the insides of spaces inside the body, and when this flagellum kind of undulates and whips back and forth, it pushes water, and as it pushes water, water gets pushed away from the surface, creating a partial negative vacuum that helps to draw water in from the outside of the sponge body over here to the left, and water flows through and through the inside of the chamber right here. Alright, so that gets water circulating through it, and of course if water circulates through, that's going to bring in oxygen, which can get close to all the rest of the cells, and so these cells can absorb the oxygen from the environment. More importantly though, the choanocytes, with this collar is actually a little filtering apparatus. There are small gaps and spaces that are coated in mucus, and so as food travels through, it gets stuck on here, and then food can be captured through phagocytosis by the main body of the choanocyte. And then the choanocyte can use that food for itself, or it can pass on those food vacuoles to other cells that can distribute the nutrients around to everything else in the sponge. Alright, so third type of cell, pinacocytes. Pinacocytes are just the very flat cells that form the outer surface of the sponge body. They're the equivalent of epidermal cells, and they superficially look like simple squamous epithelial tissue that you guys studied in vertebrates. All right. So they just kind of provide that outer surface. No other major functions that we're aware of there. All right. The final cell type are cells called amoebocytes, and they really do live up to their name because that's like amoebas, right? Remember looking at amoebas last week, and amoebas are those cells that kind of ooze around creating pseudopodia. They don't have a defined cell shape. So here we have just one drawing of an amoebocyte up here, but you can see other amoebocytes have slightly different shapes to them. All right, so what do amoebocytes do? Well, amoebocytes are probably the most I guess you would say, kind of fluid in their jobs. That is, they can change the sorts of roles that they play. All right, so among the roles, amoebocytes are very important in helping to transfer nutrients that are captured by the choanocytes and then ooze their way through the rest of the sponge body and give them off to porocytes or pinacocytes so that those cells get the nutrients that are being captured. All right. Another role that amoebocytes have is that they can actually undergo the process of meiosis and form themselves into egg cells or other reproductive structures. All right. The choanocytes actually can sometimes do that too. Some choanocytes will also undergo meiosis, and because they're already equipped with a flagellum, and if you reduce the number of chromosomes in half, guess what? You can detach one of these that is now haploid, and it becomes a sperm cell. So they can reproduce sexually, as well as some asexual methods of reproduction. All right. The last thing amoebocytes are known for doing is that they help with a lot of just the internal structure of the sponge itself. Here we see over on the right side an amoebocyte that is secreting an item known as a spicule. All right. Spicules are sort of like an endoskeleton kind of structure here. They're fine little crystals made out of different materials depending on the classification of the sponge, and they also have different shapes and sizes. I'll show you some of those in just a second. And they provide kind of that rigid framework, and then kind of secreted around it is a gelatinous material referred to as mesohyl. And that mesohyl is sort of like concrete, and the spicules is like a reinforcing rebar that gives it a little bit of strength. So let's actually take a look at some spicules here. Alright, so this is actually from one of our own slides, and you'll be able to look at these through a microscope, and you will see that they have different shapes. Many of them are kind of three-pronged, but different lengths to them. Others are simple needle-like structures. You can see a couple here and here, and others have even more complex shapes. Okay. Here's another picture. This is one I just got off the internet here. So this one is, you can see, got 6 rays on it. So really kind of pretty if you really think about it. All right. And then here's just, again, a brighter range here. All right. Now, one thing you can get on this graphic is that there are also different materials that spicules can be made out of. Most spicules are what we call calcareous. So calcareous simply means that they're made out of calcium carbonate, a material very similar to that of the shells of a lot of other marine organisms. And indeed, that becomes kind of the basis for classification of an entire group of sponges called the calcareous sponges. All right. Other sponges use silicon dioxide, and they make what we call silicious spicules. Silicious spicules, kind of like the one in the previous slide, are found in an entire class known as the hexactinellida, also known as the glass sponges. And I got some pictures I want to show you those towards the end of the presentation here. All right. We also see silicious spicules in a third group called the Desmospongiae, and you can see these are even weirder shapes, very kind of elaborate kind of forms right here. All right, and they belong to a class called the Desmospongiae. And the Desmospongiae are also interesting because in addition to these hard spicules that are, again, arranged kind of like just skeletal elements, but they also reinforce their body with a protein called spongin that you can see right over here. So spongin is very much like collagen, Right, so remember collagen from connective tissues in vertebrates. So this is very much the same type of protein, just some very minor chemical differences. And then the spongin can be arranged in this very intricate web-like configuration here to create a kind of a softer, more flexible version of a skeleton. And we'll see how kind of that can be arranged as we go through again a little bit more about some of the different shapes and types of sponges that there are. All right, let's actually move on to that. All right. So when it comes to sponge body forms, there's kind of three key forms. All right, the most basic body form is something called an asconoid body. All right, so asconoid sponges. Now, this is not a taxonomic classification. This is just a term we use to describe the basic structure of the sponge. And it's pretty simple. It's a very, very small, usually, about, a quarter of the size of your pinky finger kind of diameter body right here. So they're really, really tiny. Thin, about 1 or 2 cell layers in thickness right here, where the body simply has numerous pores on the outside that leads into a single central body cavity. We call this a spongocoel. So you can see that right here, spongocoel. All right, so that's the central cavity where all the water kind of flows into. Here you can see this picture, water flow coming in through porocytes. And again, all of this is being driven by the beating of flagella of the choanocytes lining the inside of the spongocoel. All right, as all the water gets drawn in, food gets filtered out, and then waste products can be excreted into this water as it's flowing away, and then all the water is expelled out through a large opening at the top of the body. This is called an osculum. All right, so you can see that this asconoid sponge, again, is really just a very, very simple sac-like body. Nothing very complicated about it. All right, and so it's able to thrive in environments where there's good water quality. And you see these in both freshwater and in saltwater kind of habitats. All right. The only problem with the asconoid body sponge is that it does limit the size of the animal because with only one chamber where water is able to circulate against the surface of cells and then water circulating around the outer surface, all the cells need access to oxygen. And so this configuration makes sure every cell is in direct contact with this life-sustaining fluid. Right? But you can't get very big or very thick. If you add more and more layers of mesohyl and try to bury cells into the inside, suddenly cells are not able to have direct access to oxygen and other dissolved minerals and things that it needs to survive, so they can't get very big. Right? But there are bigger sponges out there. Why is that? Well, you start creating folds in the body. And so we can go to the next type of body, which is called a syconoid sponge. And this is just a partial illustration of a cross-section through one of these bodies. And all you do is that rather than having one central spongocoel, you still have one. It's kind of indicated in this lower right corner down here where you can see it's labeled spongocoel. But what happens is that you create these kind of side branches from the spongocoel, what are referred to as radial canals. And so these are like little finger-like projections that are also lined by the choanocytes. But then among these sort of fingers that stick out to the side, that creates spaces along the side. And spaces along the side are called incurrent canals. And so the animals are really, all they're doing is creating a lot more surface area for choanocytes to exist in. And by folding it around, you can actually now create kind of a larger body structure by staggering the placement of these radial canals around a central column. And so you can get these kind of larger, more robust sponge bodies. But the basic kind of feeding and water flow patterns are still the same. Water flows in through gaps between the radial canals called dermal ostia. It really leads into a narrow chamber that is covered in smooth pinacocytes, and then there are gaps in the sides of the radial canals. These are no longer necessarily formed by porocytes, although we could designate any cells along these edges as kind of functioning in that capacity. But these openings are referred to as prosopiles, and so as water flows in through an incurrent canal, it goes across a prosopile, and then it goes into the radial canal, which is lined by the choanocytes. Now here's where, again, food is collected and everything like that. Whoa! Okay, that was unexpected. There. I didn't need to zoom in quite so close at that moment. All right. Water then flows along the radial canal, and eventually it goes through an opening from the radial canal into the spongiceal. That opening is labeled down here. It's called an apopyle. Right, and then from there, water flows through that spongiceal, and it goes up the column, and then it goes out, again, a large opening, what we call an ostium. All right, so this is actually the kind of specimen that you're going to spend the most time viewing in lab, so I actually do have a preview of what you're going to be looking at. All right, so here is through a longitudinal section of one of these siphonoid-type sponges. And again, you can see I've labeled up here in-current canals. Most of them are a little bit off-centered from where they made the slice, but here you can see a definite opening that's going into one. Right, and then you can also see here's the big spongiceal space in the middle right here, and here's the osculum. This is where I probably should have the arrow going the other way so you see that water is actually flowing out through this hole over here. And then you can see openings from the spongiceal that lead into the radial canals right along there. All right, so what we're going to do, we're going to actually take a little bit of a closer look at these in-current and radial canals so we can better see some of the different types of sponge cells. All right, so I've kind of enhanced this picture a little bit. Just went in with a marker and just tried to highlight some of these things to make it a little bit easier for you to know what you're looking at as you examine. Sorry, I'm going a little crazy there. No! There we go. All right. So here again we see kind of this opening, what we call an apopyle, from our in-current canal going across into the radial canal over here. Again, I kind of labeled these porocytes, but these are not tube-shaped cells. They're just kind of guarding the sides of the opening between the two. All right. Now, how did I know that this side over here was the in-current canal? Well, if I take a close look at the cells on the very surface of this space, you can see that they're kind of flat, thin cells. So these are the pinacocytes right along in here. And some more over on this side that I didn't highlight, but okay. But where you can see all of those kind of flat, thin little cells on the very surface, that lets you know that you're looking at an in-current canal. Right? On the other hand, I go over here and I look at this space, and I look at the cells along this side. Instead, all the cells are kind of arranged. They're very similar to columnar-type cells, but these are the actual choanocyte cells. They're all labeled. Here's a cluster I just kind of highlighted over here. All right, so you've got, again, the main cell body, little collar, and you can't really see the flagella, but the flagella would all be waving back and forth along the sides right here. All right. Now again, we can also see some irregularly shaped cells, so these are going to be our amoebocytes. I got one there, I got another one way up here in the upper right. You can see that one over there. Alright, so you can't actually see and distinguish all of these different cell types. I know it's a little more chaotic than some of the illustrations are, but you know, that's biology. It never looks quite as good as the real picture. Alright, now, to just kind of wrap things up here, we've got our third and final type, the leuconoid sponges. Now, these are the sponges that most people are kind of familiar with, because these are like the large, old-fashioned bath sponges. Back in the day before Dow Chemical created polyester and polystyrene and all these synthetic fibers that they were able to actually configure to create the modern synthetic sponge. If people wanted a sponge, they actually had to go out, people had to go out and actually collect these animals and cut them off of the sea floor, where they would produce these really large, elaborate bodies, and then they would dry them out and rinse out all of the kind of biological material, all the proteins and stuff like that, except for the spongin protein, which is what you're seeing here. All right, so these are just the complex fibrous skeletons of large sponges. So this is part of a body plan we call the leuconoid body plan. I'll give you a better idea of what that looks like. Let's just go to a simple drawing right here. All right. So rather than creating more surface area by creating these little finger-like radial canals that stuck out sideways, instead, what started happening is that some sponges began to evolve a series of open spaces or chambers that were lined in choanocytes. So these flagellated chambers, are also called feeding chambers, were just kind of pockets that were found along the pathways from canals that come in. So again, we have in-current canals and what we call dermal ostia. And then the canals would branch through the rest of the body, but it was always a relatively short distance from a canal to a series of cells and mesohyl and spicules that are all kind of packed into a space there. But then the choanocytes would simply beat their flagella, draw water in through these canals, collect the food, and again there would be amoebocytes in the mesohyl that would then distribute the food everywhere else. But by having this fine network of canals permeating through the sponge body, again, no cell was too far away from sources of oxygen and other essential ingredients for life. Eventually, the canals would merge back together into a series of excurrent canals, and then all excurrent canals would go to a single osculum leading to the outside. But the interesting thing about this body plan is that rather than having a single central spongiceal, you could have multiple excurrent canals leading to multiple oscula, and the sponge body could grow very large and across a wide variety of surfaces. All right, so some examples of these kinds of leuconoid body plans. So here's something called a mucus sponge. This is actually found here on the coast of California and other parts of the west coast of North America. Sponges often grow in shady places underneath rocks because they don't have to compete with algae and other stuff for access to light. These are animals. They're heterotrophs, so they don't need sunlight, and so they can thrive a lot better in darker recesses. All right, so what we can see here in this picture, you can see kind of all of these little dark spots kind of pointing at along the way here. All right, these are all oscula. So these are places where water is flowing out of the body. All of the dermal ostia that are sucking water into the sponge body are even tinier pores that we can't really see on this scale. All right, so this sponge, again, is called a mucus sponge because it's kind of a defense mechanism. It exudes mucus when it is disturbed by other animals that might be crawling on it and attempting to kind of feed off of it. The mucus contains mild toxins and things that just make it kind of taste bad. One of the ironies about sponges is that they are non-mobile animals. They have absolutely no muscle tissue, no nervous tissue, so they can't move. Their defenses are purely based on kind of chemical defenses. It's also why this one happens to be very bright orange. It's kind of a signal to any animals that are capable of perceiving color that I stand out in the environment. I don't taste very good. That's something we refer to as warning coloration. All right, here's another local variety of sponge. These are called purple vase sponges. And they're actually tiny little sponges. These are barely the size of the tip of your pinky finger. And they tend to kind of grow in these masses right here. All right, so the large holes, again, are oscula, where water is flowing out. All the water is being sucked in along microscopic pores along the sides of the body. All right, and then you have some of this just sheets of the sponge material in here. They don't always have to organize themselves into these little vase-like structures. All right, so again, you can get much more larger, more complex kind of bodies right there. All right, now the last thing I wanted to share with you, again, are these really unique sponges that are called glass sponges. And glass sponges are found in the deep ocean, places where there's not a lot of strong water movement, and so they can create very elaborate internal skeletons using those siliceous spicules that are kind of woven together. Look down here in the lower right, you can see the intricacy at which all of these little silica mineral spicules are interlinked to one another in this more complex pattern to create this delicate but surprisingly robust arrangement that creates this vase-like kind of body that, again, there's thin layers of living tissue that cling to the surface of all of this and are collecting food particles and oxygen, and water is simply flowing in through the sides and out through the top again. So these are very unique and very delicate. I wish we had an actual specimen for you to see in the lab. All I've got are some more pictures up on a poster, but that's about the best we're going to be able to do for you. All right, so there is a nice introduction to the sponges, and later in the lab you will be getting an opportunity to watch a full video that is going to talk a lot more about these animals and actually show you some really cool video footage that demonstrates just, even though they just kind of sit there and don't look like they're doing anything, they really are living and active animals. So thanks for joining, and we'll see you in class.